dear friends, so our topic of discussion today is non-homologous end joining, or in the short we can say it's NHEJ. So let's see how it occurs in our cells. It actually happens in the case of double strand DNA breaks. That is, when both the strands of DNA gets broken and the molecule actually separates. Now our body has to join both these strands. This is our intact DNA molecule. This is strand first, this is the other strand. These two strands are antiparallel to each other. Now let's say that the double strand break has occurred. So both the strands of DNA have broken. These are the overhanging ends. See, this is one side of the molecule and this is the other side of the molecule, and some nucleotides have been lost from this side and some nucleotides have lost from this side. This loss of nucleotides actually is known as resection. This is resection. To repair this kind of breaks, our body first of all recruits a protein which is known as Q7080. It is Q7080, which is a kind of heterodimer. So this protein comes and gets bound to the broken ends here. It's a ring-like protein. It comes and joins to the DNA molecule near the broken ends. After joining here, this protein recruits one more protein which is known as PKCCS, that is protein kinase C catalytic subunit. So this protein kinase C or PKCCS comes here and gets bound to the broken ends. So these are the broken ends on the both sides. So PKCCS comes here and gets bound to the DNA molecule near the Q7080 at the broken ends. After binding to the DNA molecule, this PKCCS gets activated. It is a kind of kinase protein, so we all know it can phosphorylate the other proteins. So after getting activated, this protein can phosphorylate other proteins now. After this, one more protein gets recruited here, that is known as Artemis. So Artemis protein also comes here and joins to the broken ends. Now we know that this protein PKCCS has become activated after binding to the DNA molecule. So now it phosphorylates this Artemis protein. After getting phosphorylated, Artemis becomes activated. This is actually it's a kind of endonuclease. So after getting activated, this Artemis protein can now remove the nucleotides from the DNA molecule. So this protein now makes the nicks here. Because it's an endonuclease, it can make the nicks inside the DNA strands. So it makes the nicks here and removes the overhanging ends of the DNA molecule. So that ultimately we have got the equal ends now. That is, all the overhang portion, this portion gets removed with the help of Artemis, which is an endonuclease, and we have the portion of the DNA molecule without any overhanging ends like this. Now these two strands have to be joined to each other. These two broken ends of the single strands now have to be joined to each other, and this joining is done with the help of ligase 4. This ligase 4 actually needs two more proteins. First is XRCC4, that is X-ray repair cross complementing protein 4, and the other protein is XLF, that is XRCC4 like factor. So with the help of these two proteins, XRCC4 and XLF, ligase 4 joins these two ends now, so that we have got a DNA molecule like this. We have seen there is loss of some nucleotides at the overhanging ends, these nucleotides. So this loss of nucleotides is known as resection, and it mainly occurs in this kind of repair. But this kind of repair is very useful because this is the prominent repair that occurs in the mammalian cells. This NHEJ is non-existent in E. coli and many bacteria. In the mammalian cells, it can occur in non-dividing as well as in dividing cells. In dividing cells, the main kind of repair is homologous repair. In that kind of repair, there is a use of homologous DNA template for the DNA repair. But NHEJ does not need any homologous DNA template. It can occur without any template also. But we can say it's an emergency kind of repair. It is not very precise repair, and we have got a loss of many nucleotides also. Then also, this repair is the prominent pathway in mammalian cells. So let's see the whole process once again, how it occurs. This is a DNA molecule. Now both the strands of this DNA molecule have broken. This kind of breakage is detected by Q7080, which is a heterodimer and a ring-like protein. This Q7080 comes and binds to the DNA molecule near the broken ends. It recruits a protein kinase C catalytic subunit, which comes and binds to the broken ends. And one more protein, that is Artemis, is also recruited, that also comes and binds to the broken ends. After binding to the DNA molecule, protein kinase C gets activated and it phosphorylates the Artemis protein. This Artemis protein is actually an endonuclease which removes the overhanging ends from the DNA molecule, both the strands. Now we have got the DNA molecule like this, where the overhanging ends have been removed. Now both the ends of these strands, that is first strand as well as the second strand, gets joined with the help of ligase 4. This ligase 4 enzyme needs two more protein for its proper function, that is XRCC4, X-ray repair cross complementing protein 4, and XLF, that is XRCC4 like factor. Now DNA double strand repair gets done with the help of non-homologous end joining.